Hello, everyone. So welcome to episode number 12 of Coronavirus Conversations. And unfortunately, you'll see that it's myself. We have a guest this morning, and Josh cannot be with us because he's busy in the Gable Health Center. It's a busy time right now. So we send our best to Josh, and hopefully he can rejoin us for the next episode. So with us today is Ken Cleaver. And so, hi, Ken, and thank you for joining us.
Good morning.
So, Ken, you're an Albright alum, correct?
That's correct.
Class of what?
1968.
1968. And what was your major?
Chemistry.
Chemistry.
And in those days, there was not even a biochemistry course, let alone a major, which of course there is today. Things were a bit different then. I don't want to say primitive, but certainly different.
Right.
It was the Vietnam. It was the Vietnam era, and there was a lot of high emotions going on all over the country, and there was a limited amount of that at Albright, but not a tremendous amount.
Okay. Yeah, I've only heard stories, right? My dad was in the Marine Corps in Vietnam, so I hear some stories from him. He enlisted straight out of high school. The idea was that if you enlisted, it was a shorter tour than if you were drafted. It was a shorter tour if you were, if you enlisted versus drafted. So he went almost straight out of high school. It was overseas for about 15 months. So yeah, I've heard a few stories. It was an interesting time in our country for sure.
Yeah, well, one of the maybe the most exciting things that happened for many of us in my class, and it happened to me. Of course, we had graduation on Sunday, which was typical. I guess that's still when it happens now. And a lot of us got our draft notices in the mail the next day to report, and that included me, to report for in the induction physical at Fort Indiantown Gap.
That's near Hersperger area, correct?
That's correct.
Yeah.
And we had a mini class reunion that day, because there must have been about 40 of our class that were all there on the same day. So we all got, we all had a visitation that day.
Wow.
Yeah.
Well, on a lighter note,
lighter note, yes.
Yeah. So where are you from, Ken?
Well, I was a day student at Albright. I went to Ole High School, which is just about 10 miles away from campus, and I commuted every day. I guess that's where Dr. Williamson lives.
Yes, right.
Yes, I was a class of 1964. Okay.
And did you choose Albright because you wanted to stay close to home, or what was the reason?
Well, yeah, kind of. Of course, one of the primary reason was that I was and still am a type 1 diabetic. So it was, I guess, a little bit of uncertainty about going away to home. It was a lot better environment for me to do my college at home, where I could have less distractions or whatever. So that was I guess the primary reason. And Albright was and is a good school. I'm not one bit sorry that I made that choice.
Absolutely.
And I met my wife. She's also a class of 1968, so it was a very good choice.
Right. And I'll speak to the sciences just because that's where I am, right? In the biology department. But I mean, I don't think you could get a better liberal arts education anywhere, but specifically in the sciences, because that's what I know, but the most, it's top notch. You get, you get that exposure to your professors, like one-on-one. We all care very much about education, and that's why we're here. So I'm very proud to be, part of Albright. It's, especially in the sciences. Like I said, that's what I know the most. But we're known for other things as well. So it's a good place to be for sure.
Absolutely.
Yeah. Okay. So 1968. So just a little bit of a side note, because I teach about genetics and genetic engineering and all sorts of these techniques with my biology students. Like the cloning revolution, or like DNA technology, and being able to clone genes and express them in organisms, didn't really take off until the 70s and 80s, right? So, I mean, one of the things I talk about in my classes is being able to make just boatloads of insulin in a lab, right?
Yep.
So I'm just curious, like what was the treatment for type 1 diabetes back in, say, the 50s and 60s?
Well, of course, insulin was originally discovered as an extract of beef and pork pancreas. It all started, I believe, in 1928 with doctors Banting and Best up in Toronto. And well, I actually became a diabetic in 1949. So
Okay.
at that point, it was it was quite available. But in my case, I actually got it at my doctor's office. Every time I went there, we bought insulin, the same size vial as it is now, a 10 cc vial, and it remained. I took that extract, the beef and pork extract, until about the mid 60s, when they started to come out with human derived insulin, and then later on I guess somewhere in the 80s, they started to produce the more synthetic versions of insulin. So yeah, insulin has changed quite a bit during the period of time I've been on it. I'm coming up on 71 years next month of taking insulin, so it certainly has meant a lot to me.
How did they prevent rejection, right? Because you're introducing beef and pork insulin, right, which is a foreign protein. Did they have to do anything to prevent rejection of that foreign protein?
Well, I know of people that develop a lot of resistance to the insulin. In fact, one of my good friends here where I live is also a long-term type 1 diabetic, not quite as long as me, but he developed insulin resistance to the beef and pork product, and so you basically got around it by just having to take more.
Okay.
So he had to take a lot of insulin until they switched over to the human product. I was very lucky because I never developed any any significant resistance, and my dose was quite stable over the years up until the time that it was switched over. So in that regard, I'm very lucky.
Great. Okay.
But insulin resistance was well known, and that was fairly common.
Yeah. Okay. Well, obviously, that's a bit of a side note, but I'm a science nerd, so I'll admit it, right? I'm just curious.
Right.
Okay, so you graduated Albright, and then you got a PhD in chemistry. Is that correct?
No, I got a master's degree in chemistry at Temple from and my one of my lead professors at Temple was another Albright grad named Dr. Grant Crow,
Okay.
who is pretty well known in Albright circles. He also was a very good track star at Albright when he was there. So I studied under him for several years, and then I was working at the time. I then went back to Temple and got my Ph.D. in pharmaceutics.
Pharmaceutics. Okay.
From Temple Pharmacy. Pharmaceutics is that's pharmacy school lingo for manufacturing and quality control. My thesis was actually on clinical research, the clinical research process around the world, and the development of drugs.
Okay.
I happen to be working at Janssen Pharmaceutica, which is a division of Johnson & Johnson, and is also one of the lead companies in the COVID-19 vaccine research that's going on. That's when you hear about the Johnson & Johnson vaccine, it's actually being developed by Janssen.
Mm-hmm.
So I headed up the clinical research operations group at Janssen for several years. Worked on a number of very interesting drugs, which did get developed and approved. And during that time, I went back. I was going to Temple Pharmacy School part-time, and got my PhD in 1984.
Okay. Very, very interesting. Is that term pharmaceutics still around today as a?
Oh yeah.
Yeah.
Oh yes.
I'm not as familiar. I've heard of degrees such as, well, being a pharmacist, obviously, pharmacy school. I've heard of pharmacology, which is basically the study of how drugs work in the body and the mechanism.
There are three principal disciplines in pharmacy school. Actually four, because what used to be the basic degree in pharmacy, the old RX degree,
mm-hmm.
has now become a PharmD program. The doctor, that's the one discipline. Then there is pharmacology, which is still there. There's pharmaceutics, and then there's also medicinal chemistry.
Okay.
So that's the more scientific research and more pure chemistry. And then pharmaceutics, which, as I said before, is primarily deals with manufacturing and quality control.
Okay.
And then pharmacology is also a more scientific degree, which studies, of course, bioavailability, drug absorption, drug delivery, drug distribution and metabolism, things like that. That's more traditional pharmacology. But I focused on pharmaceutics, which was where I was working in actually for a long period of time.
Okay, very interesting. So I went to Bucknell for undergrad, and I had a lot of friends that were chemies, chemical engineers. So
right.
Can you just sort of educate me a little bit, because it sounds like there's some overlap to me, sort of an uninitiated outsider, right? It seems to me that there would be some overlap between what a chemical engineer does and what a pharmaceutics person would do. So what is like the overlap and what is the difference, I guess?
Well, chemical engineering, of course, you take a lot of physics, calculus, things like that. More, whereas pharmaceutics is traditionally more involved with the actual drug development process, taking a chemical or biochemical
Mm-hmm.
from the research stage through the scale-up manufacturing, and into clinical trials and then on to approval. So there's a regulatory component to that. There's also scale-up problems. You're involved with stability studies, developing expiration data, and then the clinical research process, which of course is clinical trials, where first of all you study the drug, both the safety and the mechanisms of action in research animals, rats, mice, guinea pigs, dogs, et cetera, and of course there's been a lot of movement away from that recently to try and reduce the exposure and the use of laboratory animals because of PETA and things like this. So that's one area that has changed quite a bit from when I got into the field to now, where there's more focus on laboratory and scientific research into the safety rather than using a lot of animals. There's a lot of pressure to get away from that. So, but then you from there you move into the study of drugs, both drugs and medical devices. Of course, is part of this process as well, because there's a lot of development time being that's spent on medical devices.
Yeah, that's becoming a big area right now, right? I mean, it's
just
exploding.
medical devices has been a huge area for many years. There's a lot more medical devices out there than there are drugs.
Mm-hmm.
When you think of everything that you use, from syringes to diagnostic products to lots and lots of things, are all considered medical devices. And of course, there's the safety and efficacy profiles of all of those things in medical devices as there are in drugs. So I've been
under the FDA, correct? All under the FDA?
All under FDA supervision and guidance, yes, correct. Okay. And I've actually worked quite a bit with medical device development as well as drugs.
Very cool.
Over the years.
Sure. What's your favorite drug or your favorite device that you've had the sort of the opportunity to work on, like the one that stands out that you're either the most proud of or you thought was the most interesting? What's the one product?
Oh boy.
Sorry to put you on the spot, but it just popped up.
Yeah. Well, that's there's a lot of them, because of course I'm an old guy, so I've been working since 1970. So that's a pretty long time. I guess, well, two of the drugs that I was particularly interesting to me, of course, were Janssen products, and one is the family of drugs that included miconazole, ketoconazole, and itraconazole, which are antifungal drugs. Okay. Those were kind of a specialty area of Janssen. And then also the clinical studies of the children, what we call the children of fentanyl. Of course, everybody knows about fentanyl, and it has plenty of notoriety for its narcotic properties. But while I was at Janssen, we developed the analogs or children of fentanyl that are named alfentanil and sufentanil. Now, sufentanil you've probably heard of, because that's one of the drugs of abuse that gets a lot of press because of product coming in, illicit product coming in from overseas. But sufentanil is a very, very useful drug for many types of surgeries. While that was being developed, I observed a number of cardiac bypass operations, things like that, where I was actually in the operating room while sufentanil was used as a narcotic analgesic for some of these very prolonged and very painful surgeries. So that was, I guess, one of my favorite products. Devices that I've been involved with directly include a class 3 device, which is marketed for severe epilepsy.
Okay.
That's an implant that's implanted in your chest, and it actually provides an electric stimulus to help control patients who are not controlled on traditional epileptic drugs.
Okay.
And that device has been approved for quite a long period of time now, but it's also being studied currently for use in depression and Parkinson's disease.
Okay.
So these are serious forms of those two afflictions, which seem to be responding, from what I read, in helping to suppress severe Parkinson's disease and also depression.
Sure.
So that's probably my favorite device that I was involved with.
Very cool.
And that was during my consulting career. I left industry in 1992 and opened my own consulting practice, which I've been doing still up to this day to a certain degree. I'm more or less retired, but I'm still involved with a couple of companies, and I'm still teaching a class at the University of Maryland College of Pharmacy. I taught it this summer. It's an online course, so it's not, like I wasn't standing up in front of a bunch of students, but it's in the graduate school at the University of Maryland.
Yeah, and that's kind of how I envision my retirement. Hopefully, many years from now, I don't think I'll ever be able to give it up completely. But yeah, well, that's very cool, Ken. So this is a very sort of opportune time for us to be speaking. We were chatting just briefly before we hit record that we've had the Pfizer trial results come out last week, and the Moderna results come out just yesterday.
Correct.
And before we get into that, I just wanted to kind of give our viewers and listeners a little kind of backdrop. And you had mentioned about animal studies and things like that, and those are important. I don't know if those will ever go away, right? Because they do serve a very legitimate purpose in trying to figure out if there's any, like, say, toxicity or side effect issues.
Correct.
And if there's some major toxicity discovered in a small animal model like a mouse, that's never going to make it into a clinical trial. So the idea is we're trying to prevent things that are toxic or have unknown side effects from even touching a human being at all. So I think animal models have a purpose, and I don't think they should ever go away completely. But to agree with the people that want to shut it down completely, I think we should limit their use as much as possible, right, and do it in a humane way.
Yes.
But we don't want to discover some intense side effect, some sort of toxicity issue in a phase 1 safety trial. So we can do some of this in culture, in tissue culture, with cells in vitro, like in a culture dish, and if it looks safe in there, looks safe in a mouse, then you can sort of continue down the pipeline.
Correct.
Yeah, so that's the preclinical sort of side of things. And then so Ken, so just can you briefly walk us through the different phases of clinical trials, right? So phase 1, right now, correct me if I'm wrong, are they kind of sort of combining phase 2, 3 together? Can you sort of briefly walk us through what all these phases mean, and what kind of happens in those phases, and then we'll get into the actual vaccines.
Okay. Well, there's. I'll try to make this as brief as possible because it can become a very long story. But yes, traditionally, and this is true for drugs, devices, biologics, vaccines. The first initial trials obviously called phase 1, is where uh, a potential product is given or administered or implanted or whatever to healthy volunteers. Now, this is typically up until about 20 years ago, they only used to do this kind of work in typically healthy young men. Now they were that changed about 25 years ago, where they started to include older people, women, sometimes young people, teenagers, or even babies, young children, to get a better exposure to the whole population. So they study that first for a single exposure, and then multiple exposures, because most things are used on a chronic basis. So you have to study it, first on a short-term basis, then on a longer-term basis, to get an idea of, is there going to be some serious toxicity, and you want to study that first in healthy people, because they have the best chance of recovery. Then traditionally, then once you have an idea of safety, then you move into what's called phase 2 trials.
Oh, sorry, Ken. Can I just have one quick follow-up? So these
Yep.
safety trials, how many people we're talking? I mean, it's typically not a large number, right? Like maybe
No, it's typically
50, 100.
It's typically perhaps somewhere between 50 and 150.
Right. But the idea being, again, you don't want to give it to 10,000 people right away in case there is a problem, right? You give it to a smaller number of people. Gotcha. That makes sense.
Correct.
Okay. All right.
Okay.
Yeah, sorry about that. Yeah. And then, all right. So now phase 2.
Phase 2 is when you start to, I mean, you have an idea from the class of product that you're working on, what this chemical or device would be used for. So then you start to look at given populations that have potential for treatment, whether it's a drug or a device, and you start to treat a larger but not very large group of people with the potential product for either a relatively short or moderate period of time. Again, the point in doing this is you want to get some idea of is this product going to be potentially useful? You're still looking at all the safety aspects of this. Does it cause any kind of side effects, or hopefully nothing serious, but what would the effect might be? So you're looking at both effect and safety. You can't, you don't study enough of these people. You're also might be looking at the dose of a drug, because if you're looking at a drug that may be useful for, say, rheumatoid arthritis, you have to look at the way that it's given. Do you take it in the morning? Do you take it at night? Do you take it 3 times a day? And what type of dose? So there are different cohorts of people and how you administer this stuff. So you're getting a better idea of where this potential drug or device might fit into the treatment of groups of individuals that that might be useful in. And of course, many drugs have different uses in different doses. For example, sildenafil, which of course is the parent compound for Viagra, which we all know what that's used for, has multiple uses depending on the dose that's given. Lowered, one dose is useful for treating blood pressure. The other one, a higher dose, is useful for treating erectile dysfunction. So, one drug may have many different uses at different doses. Aspirin is a very good example of a drug that's, on a short-term basis, it's very good as an analgesic, but on a longer-term basis, at a lower dose, it's useful in cardiology.
Mm-hmm.
So, a given drug may have multiple uses in multiple populations, and you learn about this over time. So once you have an idea of the profile and how a drug might be useful, then you traditionally would move into phase 3,
And
which is
if we bring this back to vaccines, Ken, so like in a traditional sense, and this I guess is why typically this has taken well over a decade, right? Because you do
yes,
say a phase 2 dosing trial, you're maybe monitoring the immune response generated by your vaccine, and then that'll inform, I guess, how you set up phase 3 then,
yes,
in terms of
and
dosing and
you,
things
I
like
mean,
that.
traditionally, yeah, phase 2 may take several years
Yeah.
to study, because you're looking at a whole variety of different things. Now, in the case of the COVID vaccines, because there's such a criticality of medical need for this, phase 2 and 3 have kind of been squashed together.
Mm-hmm.
So the phase 2 trials in the vaccine world that we're living in right now has been very much compressed. Whereas if in a more traditional sense, you would have looked at these vaccines over several years and studied the long-term benefit and learned how much the immunity boost might last. Whereas in with the COVID vaccines, this has been compressed so that it's looking like we're going to have these products introduced without a lot of knowledge about what the long-term effects are. In other words, how long the immune response is actually going to last.
Right.
And you'll hear about this in the news, when they say we don't know how long the antibodies that are built up from the vaccines are going to last. And this is just because there's such a great medical need for it.
Right. Makes sense. And I would also, like to point out to our listeners that this again speaks to the importance of the animal models and the preclinical work, because these companies, because of the animal work, have a general understanding of the immunogenicity, a general understanding of dosage, and you could sort of, have an idea. You can kind of scale up from a mouse to human. How many, how much, how many grams or micrograms of this product or whatever we gave it to, this 25, gram mouse. In general, like we can start here with humans. So again, that speaks to the importance of the animal models in my mind. So you're not flying totally blind, right? Going into the human work.
No, you can, based on the animal response, you have some predictability
Right.
In there that you can hopefully gain, a lot of information from those early animal trials to know what's going to happen in man. And that's why, different drugs get studied in different animal models. For example, drugs that are useful for skin, the study of skin get a lot of attention in pigs, because the skin of a pig is very similar to the skin in man. Both they have an epidermis and a dermis, which are quite similar in man. That's why different animal models are studied for different types of drugs for the eventual use. I mean, sometimes you'll study things in sheep, or you'll study other drugs in dogs, things like that, because the metabolism and the musculature patterns in different animals is similar to man. So the type of animal that's used for these early studies will vary from product to product.
Makes sense. Okay,
Yeah.
so let's move on. So phase 3, so that's typically considered the efficacy, right? So
Efficacy.
how good is this damn thing going to work, right? So
Yes.
yeah, so right now, so for phase 3 right now, my understanding is that all these different companies have recruited approximately 30 to 40,000 participants from around the world. I think the Pfizer study was in multiple countries, including the United States, to accumulate data.
Correct.
And sort of a gold standard trial, right? They set up these double-blind, placebo-controlled. So can you explain a little bit about? So for a lot of these, they were setting up double-blind trials, right? The person
Correct.
giving the vaccine, giving the shot, doesn't know what's in the vial, and nor does the person receiving it. And there's also a placebo group that essentially gets like a dummy dose, if you will. And so what's they call that the gold standard. So it helps reduce bias. And my understanding is that it also reduces the risk of people maybe acting differently in a behavioral sense. So, I mean, if somebody knows they're getting the actual COVID vaccine, they might act a little differently. They might, act a little bit, a little more brazen, right? Or behave a little bit differently. They think they're protected. Whereas if
Less
somebody knows,
responsibly.
yeah, exactly. And if somebody knows that they're getting the placebo, maybe they don't go out go to the house anymore. And we, the companies, of course, counsel all of these people, and they educate them as part of the clinical trial in terms of like, but it's a very odd setup, Ken. Just between, just between people chatting here, it's such an odd setup because at the end of the day, right, the companies need to have people get infected to know how good the vaccine is. So you educate
Yes.
people on how to behave properly, like mask wearing, distancing with respect to the COVID pandemic. Or, back when I was in graduate school, we were working generally on like HIV vaccines and immunology.
Mm-hmm.
And of course, back in those days, I mean, companies would educate people on proper, safe sex, sort of practices and wearing condoms and all that. So you educate the people, but I guess at some level, that just at some point, somebody's not going to do the sort of right thing or ethical thing all the time. There's going to be a breakdown in decision making, I guess, and so there's a potential risk of an infection event. And you have your placebo group and you have your vaccine group, let's say, and you look at just how many people got infected or didn't get infected, right? So what? Okay, so we have phase 3. So what is typically so Ken, speak a little bit to the current results that just came out, right, in the press, and I guess what's the next step after that, in your mind?
Okay. Well, now both the Pfizer and Moderna results that have been reported are you've probably heard the term. These are preliminary or early results. Now, how this is set up? It's set up when they write a protocol for these large studies and submit it to the FDA. There are statistical criteria when you run any clinical trial, any large clinical trial. You build statistical points of time in the population where you will look at preliminary results or results perhaps it's halfway through the enrollment period. And you may have heard in these trials that FDA, when the protocols were first written and submitted, they were going to look at results after 1 month. They were going to give the people the 2 doses of the vaccine, and then they were going to wait 1 month and look at results. Now, FDA changed the criteria after the protocols were submitted and they said, oh no, 1 month is too soon. We want you to wait an additional month. We want you to wait until 2 months have passed until you look at results. That's one of the reasons why, results are a little bit delayed from what was first predicted, because originally you'll remember, of course, it's, all part of the political stuff that's happened recently. They said, well, we're going to have results in October.
Mm-hmm.
Well, the original protocols were written that way, that they could look at those, but FDA changed the rules. They added a month to this to gather additional data for making sure that the results had more certainty to it. So that was a change in the overall regime of running the study. So anyway, so we've reached those 2 time points where both companies were able to look at their results based on the original protocol at approximately halfway of the population. Now, they both reported these results. One is 90%, and the other one is 94.5 or something like that. That's based on the first time point. Now, they're both going to wait for an additional period of time until they get to look at the full complement of patients, the full group that they've studied,
Right.
to come up with a more definitive number. So those 2 numbers that we have at the moment are likely to change because, with a bigger population,
Mm-hmm.
those results, 90% may become 83% or 87% or something like that because it's a bigger population. So those numbers that you've heard right now are preliminary results based on partial populations.
But it certainly
So,
sounds promising, right?
but they both sound very promising.
I mean, I would rather start at 90, 94 than, let's say, 50, right? Especially what we need right now, right?
Well, yeah, and what you typically get, or what's not unusual with the flu vaccine, is that that percentage changes every year. A couple of years ago, the percentage of the both the potency and the effect of the flu vaccine was somewhere around 30%, whereas then another year it's 70%.
Right.
So that's why that number will be different from year to year. With the flu, it's a different strain that's
Right.
causing the flu and things like it. And the flu vaccine, of course, is a guess at what the strain that's 6 months off. They develop a vaccine based on what happens in Australia,
Right.
in the southern hemisphere, and then they build a vaccine or develop a vaccine around that strain or combination of strains that's out there. That's why the flu vaccine varies so much from year to year, because it's only meant to be a 1 year treatment. That's and we don't know at this point how long this COVID vaccine is going to last. I mean, this may possibly become another yearly event that happens, because we just don't know at this point. We don't have experience with this vaccine to know what's going to happen.
Right. And this is something that just came up yesterday in my microbiology class. We're actually talking about the immune system and vaccines in that particular class period. And
Yep.
There's sort of 2. To go back to the flu vaccine. Yeah, I mean, you said 1 year it's 30%, another year it's 70%. So there's 2 major goals of a vaccine. One is to prevent infection altogether.
Correct.
People call it sterilizing immunity. So basically, you're exposed, the pathogen enters the body, but it doesn't replicate. It doesn't go anywhere, and that's completely preventing infection. And then the other goal, if you can't achieve that, then you try to reduce disease burden. You basically try to make the disease course weaker or somehow lessened, right? So the person doesn't get as sick. Now, for flu, that 30 to 70% number, that's from what I can gather, just from reading about this over the years, is that is mainly looking at the former, meaning that it's looking at actual infections, right?
Yes.
But I always tell people, even if the number advertised is, say, it's 40% efficacious, still get it, because the disease course is likely to be less severe. Even though you get infected, the disease course is less severe, which is really critical for really young patients and really older patients. And so again, you might get infected, but the disease course is lessened. And I think, Ken, that is likely to be the case here for coronavirus. There's a lot of evidence to suggest that, because there are five sort of circulating coronaviruses that are considered sort of like seasonal coronaviruses, and studies have shown that people can get reinfected with those, right? You don't have like a lifelong sterilizing immune response. And it depends on the pathogen. It really does. So for example, people that survived smallpox back in the day, right?
Yes.
You were completely sterilizing immune the rest of your life. There was no sign of infection. As soon as you got in your body, it was like pretty much shut down. Whereas these coronaviruses, for reasons that people are still studying, and it's going to be interesting to find more out about this, is from the seasonal coronavirus field, people definitely can get re-exposed and reinfected, but they do not seem to have as severe of disease. And so my suspicion is that even with these vaccines, is that it likely will prevent disease, which ultimately I think is the number one goal right now. But if it doesn't, if it's not sterilizing, that means somebody can actually get infected and have the virus replicate, and potentially that person can transmit it or spread it. And so in my mind, the key to all of this is to have as many people as possible vaccinated to achieve what people would call a herd immunity, so that,
Correct.
so that if you get exposed, you don't get a disease. Because somebody that's vaccinated in this scenario that I'm discussing could still potentially spread it, just not have severe disease. So only time is going to tell. I suspect that it's not going to be sterilizing, but I suspect that it will prevent disease. So for example, in these early trial results that came out, they had a certain number of infections. Let's just say hypothetically, just because it makes the numbers easier, like 100 infections, right? So in this interim analysis, there was 100 infections. Well, that means that people showed symptoms, and they got a test to confirm that they were positive. Well, those numbers mean that essentially about 9 people, or about 90 some people that were infected were all in the placebo group, and a couple of people were in the vaccine group. And so what I'm interested to find out, Ken, when they release these results, is of the people that got infected. Okay, so a few, right? Remember, a couple of people in the vaccine arm got sick and were positive. I'm wondering if the severity of the disease, if the symptoms were lessened versus the placebo group. They didn't release any results on that yet. That part I'm most interested to learn about.
Yeah, and what you have to pay attention to in this analysis, or the results of what you're talking about here, is the ages and the medical conditions of those people that got sick. Are the ones that did get sick, were they the older, more infirm group, or were they younger? So you have to look at their immunity, their immunity class, or whatever, if you want to call it that, in each group. Are there and that's what you would call if the one group, let's say the 5 people who got the active vaccine, were all 65 plus or something like that, who, normally have a lower normal immunity, then
Right.
that would be even more interesting, because then that would mean that, this was maybe even a better protection than, if it was all in one age group, because the breadth of the people that were included or that were studied may differ in the two groups. And that's why you have to really look in the weeds, the real detail of these studies, to see what population, who got what.
Yeah, it's a really good point. Really
Yeah.
good point. Okay, so
So now
this has been
I'm gonna have to take. I just got to signal my because I'm talking on my cell phone here. My signal is getting lower. I'm gonna have to go take a 1-minute break and go get my cord and plug in. So
all right, then we'll wrap up. Okay, sounds good.
well, I don't mean to wrap up, but
Well, no. I have a meeting shortly anyway. So just the last piece I wanted to go over was kind of like the distribution piece, and then
yes,
we can kind of wrap up. So thank you.
Very important.
Okay, go get the cord.
Okay, all right. Give me a minute.
Okay, so we're back to recording again. Yeah. So.
Well, you don't need to see my mug.
All right. Right. So yeah, in the few minutes that I've left, so I was really hoping that you could, given again your expertise, just speak a little bit to the what happens next. So they finish phase 3, let's say. Let's hope and assume that the safety monitoring is still, everything looks good there. And I just also want to stress to everyone listening that they never stop assessing safety, even after a vaccine is released. There's a vaccine safety monitoring service or system that that is still in place. So they never stop looking at safety throughout all of this. Right. So let's assume
that's true for every product and every drug, every device that's out there. There's a national monitoring system that's used for all of these things.
Right, which is important for sure.
Yes.
Because correct me if I'm wrong, but wasn't there a famous example? Was it Vioxx, Ken, that was actually licensed and it made it through all of this, but then it was later pulled?
Oh, Vioxx is maybe one of the best known examples, but there have been many products that have made it to market and were later taken off the market for safety reasons. Yes, that's not at all uncommon.
Sure. Okay. Right. So
Vioxx is a good one. That's an excellent example.
So again, given your expertise, let's assume that it makes it through, it goes to market, right? So so what's like the final piece? Like, how do we get this to like a doctor's office or whatever?
Okay. Well, what you've probably heard, because everybody's talking about it, is that the two products, Moderna and Pfizer, both vaccines, are based on the same technology, messenger RNA, which are almost brand new technologies. There's never been a vaccine out there based on this technology. So this is a whole new therapeutic area that's out there. So companies do not have a lot of experience in developing these things. So there's very little information that's out there on real-time stability of a product. That's storage and distribution. So the two different products are significantly different in that the Pfizer product, at least what they're saying at the moment, is going to require very unusual storage and distribution conditions. It's going to have to be stored basically in ultra cold freezers, and will require dry ice to distribute this stuff. It has to be kept extremely cold because its stability is quite limited. Now, that's going to cause, in theory, that's going to cause a lot of problems because there's large metropolitan hospitals,
Right.
in major cities are better equipped to handle this because they have the equipment, the storage capacity, and the deep freezers to handle these kinds of things. Whereas somebody like Dr. Williamson,
Mm-hmm.
who has a has his practice, and even at Albright, I would bet that you don't have a deep freezer there.
Actually, we do, Ken.
You do? Okay.
We have
Oh, well,
two. We have two. So Dr. Williamson can have a couple of our shelves. Yeah, we have a couple of shelves we can spare. No, we do. Luckily, we have some minus 80 degrees Celsius freezers, and that's mainly for some of the work that I do, some of the work that Dr. Samuelson does in our department. But the work that I do with my viruses, we need to store the viruses that I work on in my research. We need to store them ultra cold. So.
Well, that's fantastic.
Yeah.
So okay. Well, that's very lucky.
Right.
However, what Pfizer developed to distribute these things is, and you probably heard this term, that they're using portable freezers, which are dry ice shipping containers.
Mm-hmm.
But they are designed to to hold either 1,000 or 5,000 vials of this product. So, if Albright would want to, or would be receiving these things, I've not heard, and I don't really know how they're going to distribute a number of these vials to a smaller facility. Let's say Dr. Williamson's private practice,
Mm-hmm.
who I would guess that he probably does not have one of these freezers in his office, and he probably does not have a large amount of space for storing these things. So the logistics of getting these things to private doctors and small pharmacies is going to be a challenge. I'll leave it at that. I heard the show on 60 Minutes the other week about distributing these things.
Mm-hmm.
It's going to be challenging. Now, the Moderna product, on the other hand, is that this just came out yesterday. They're saying that is only going to require probably regular freezer space, which is fortunate.
Right.
But again, I haven't heard anything about how they intend to distribute these things and what kind of quantities. So that the distribution of both of these products is going to be challenging. Just getting and plus the fact that you have to have 2 doses of each one. I believe it's 3 weeks apart.
Right. Mm-hmm.
So that's going to take a lot of coordinating. Let's say at Albright, there are, how many students do you have at Albright? I hesitate to, I should know this number, but I don't.
I want to say it's somewhere in the maybe 1,500 range.
Okay, so let's say you would want to give this to 1,500 Albright students. That would have to be, scheduling of 1,500 students at 3-week intervals is going to take some coordinating, right on your end. Then plus ordering the supplies,
right,
getting them in, storing them, coordinating, the days that they get them. And, the Pfizer product, for example, they say, once you thaw an individual vial,
mm-hmm,
you're only going to be able to use it for 5 days. So there's going to have to be a lot of coordination and timing, and there's going to have to be some very rigorous somebody in charge of that very thing, because the one thing you don't want to do is you don't want to give somebody a dose that's been expired, that has been exposed for too long.
Right.
So that's going to have to be very carefully monitored. And in a small doctor's office or a small pharmacy which is dealing with the public, it's going to be a real challenge.
Yeah, for sure. That's a good point.
Whereas what I've been hearing about the Johnson & Johnson product is that it's a single dose, and that it may only require refrigerated storage. So, I mean, that in itself is a very significant difference between the products.
Right. And I think maybe that's a silver lining in all of this is, luckily we're not relying on a single company or a single platform,
Well,
to
yes.
reach the finish line. We have, at this point, I think I checked the other day, I think there's 10 or 11 vaccine platforms from various companies that are in late stage clinical trials. It just so happens that Pfizer and Moderna were the first out the gate with some early results, but we'll start hearing results from some of these other companies within the next month probably. So
Yes.
again, like you said, maybe if they're all shown to be roughly the same efficacious wise, which let's cross our fingers. Perhaps in terms of distribution, I mean, I'm just spitballing here, but maybe, the ones that require ultra cold freezers, maybe they go more to like urban areas with
Yes.
better hospitals, equipment and lab equipment and all those things. And maybe just the refrigerator versions go to, rural parts of America with small doctor's offices and just a refrigerator. So again, I mean, you can imagine a scenario like that. But I'm glad you're bringing up these issues because, in order to get to the finish line, something as simple as whether or not you have the right freezer makes a big difference, and it's something that people probably don't realize. So thanks for bringing that up.
Okay, and one other thing I wanted to bring up as well.
Mm-hmm.
The manufacturer of the actual manufacturer of these products. Now, a lot has been talked about that Pfizer is manufacturing a lot of their product. I don't know what percentage of their total product at one location in Germany. Okay, it's a very large facility.
Okay.
Which is, from a manufacturing point, is very good, because the lesser number of sites that you manufacture a product at increases the uniformity from batch to batch.
Okay.
Whereas Moderna, which is a smaller company, certainly smaller than Pfizer. Pfizer's as we say in the industry, the big gorilla in the industry. Moderna is a smaller company, and they're going to be making their product at a number of different facilities. They don't have a large manufacturing facility, so they are utilizing several different manufacturing locations. Whenever you're forced to do that, you introduce variables into product manufacturing. You're using different equipment, different people, different handling, and the number of the amount of batch to batch variations that can occur in making any product, whether you're making tablets or whether you're making vaccines, can be exponential in their differences from one location to another. So that's also something that needs to be carefully monitored and watched, as to whether product that's made at plant 1 is going to be very close or identical to a product that's made at plant 2. And the more locations that are involved makes that difference, increases the chance that you're going to have, one batch of vaccine from, let's say, plant 2 that may be less potent or less efficacious than product made at plant 1. So, as good as the Moderna product sounds, because of the less requirement or lower requirements for storage, the fact that it's made at a different plant than plant 2 may introduce differences in efficacy from one batch to another. That's something that also has to be kept in mind and followed very closely.
All the details are kind of maddening, Ken, I have to say.
Well, those are the kinds of things that go into product development. That all of these things get looked at in quality control and in long-term storage stability,
right,
and things like that. These are all things that can all become very important.
okay. Well, Ken, I just want to say on behalf of Josh, who again, unfortunately couldn't be here for today's recording, just want to say thank you very much for coming on. Just so everyone is aware, Ken always emails me after every episode, and so it's good to have somebody out there that I know is listening. We get some good questions, and he shares articles with me and all these things. So it's good to finally have you on and for sharing your expertise with us on this. So we'll keep we'll keep chatting, and we'll keep monitoring the results as they come out. So
well, I'll keep sending you articles,
yep,
as long as you don't tell me to stop.
It helps keep me up to speed in case I miss something. So thank you. All right.
Okay. All right.
Okay, Ken. Well, thank you again, and we'll chat soon. All right. Stay safe.